Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog. Good evening, I'm Yvonne Stapp for Science for the Public, and welcome to Contemporary Science. Tonight our guest is Dr. Chris Marx. Chris Marx is an associate professor of organismic and evolutionary biology at Harvard University. He received his PhD in microbiology from the University of Washington in 2003, and he then served as a National Science Foundation postdoctoral fellow in microbial biology at Michigan State University until he joined the Department of Organismic and Evolutionary Biology at Harvard University in 2005. Professor Marx has received numerous awards, including a National Science Foundation Career Award given to young academics who've excelled both in research and teaching. He's also on the editorial board of a major journal, Frontiers in Evolutionary and Genomic Microbiology, and he's an active member of many professional organizations. And we are proud to add the Union of Concerned Scientists, whose efforts to inform the public are very well known. The focus of Dr. Marx's research is microbe evolution, and especially the context that influences adaptations within individual microbe communities and between neighboring communities. The Marx Lab analyzes such changes by means of synthetic or experimental evolution, which allows for more precise observation of very complex processes. The how and why of these experiments will be explained tonight. This kind of microbe research is highly valued today, for example, in medicine, in genetics, and in the study of evolution generally. We appreciate Dr. Marx's effort to make this area of science accessible to the general public, and it's a very great pleasure to welcome Dr. Chris Marx. Welcome.
So I'm going to talk to you about one of the particular aspects of the work that we're doing in my lab today, but it would be absolutely remiss on February 12th to not mention, it is also Lincoln Day, but this is Darwin Day. Happy 204th birthday to them both, born on the exact same day. So in terms of evolution, the particular question that I'm going to talk about tonight has to do with cooperation. And of course, there are many non-microbial examples of cooperation that many of you are familiar with. You know, most sorts of these kind of pictures are the kinds of things we've seen in nature shows from the time we were small about communities of organisms, whether they be kind of larger and more furry, down to smaller and more buzzing around, etc., that work together. But what we haven't typically realized, and even professionally as microbiologists, is microbes are not necessarily so different. And so microbial growth can also often be very public. And so if we think about how microbiologists have studied microbes from really the start of the field, is to isolate organisms, get one thing to grow all by itself, and we typically grow it in, say, a flask in the lab. And so we take away everything else that it used to live with. We take away all the structure of the environment and grow it in a well-shaken liquid environment. And yet they live in a much, much more complex scenario where most microbes live in a spatially structured world surrounded by many other microbes that are not even necessarily of their own species, that they have close physical contact. And because of this, they're not just these little kind of self-centered planktonic things that we typically envision if anyone even thinks about a microbe. They have all sorts of complex aspects that they have as interactions between them. And so I've shown pictures of a number of these things. They range from molecules that are made by individuals, but they go out into the environment. Any one of them could come back to benefit whoever is lucky enough to get it. So there are molecules called siderophores that can go out and bind things like iron in the environment and be able to make it available to come back into the cell. There are signals that are sent out from individuals and heard by the crowd. There are aspects of physical structures like biofilms, the ability to kind of make a little microbial city. You might see this happen in your fridge if you leave things in there too long. Even things like social motility, that there are actually some bacteria that can, as a group, move across a solid surface. And it's something that they can't do in the same way alone, but they can do so kind of as a wolf pack. And so there are all these various things that microbes can do that involve a public good. Individual cells are making something that is used by the total, and thus there's kind of a challenge, of course. I'm paying the bill, and everybody benefits. And that's where the evolution of cooperation becomes interesting and potentially, like I said, a challenge in terms of how that doesn't get cheated upon. And so if we think about microbes, one of the ways that they interact with each other is in terms of metabolism. And so you can have kind of a food web where the details aren't so important, but basically you can have some microbes eat some compounds in the environment, say, organic material, dead plant or animal material. The polymers in that can get broken down into monomers. These can be broken down into further products. Other organisms can use those. Other organisms use those, et cetera. And you have kind of this food chain of one thing is making something for the next, which is making something for the next, et cetera. And at one level, this is kind of cooperation. Certainly, some organisms are benefiting from the others, but it didn't necessarily evolve for that purpose. And so I like to think of this as being similar to this organism cooperating with this organism, right? There's a nutrient exchange. The first rather larger organism is making something for the second little dung beetle, but it's not for the sake of the dung beetle, right? It's a consequence of its own actions. And in this sort of cooperation, it's helping, but it's really just a byproduct. There's no conundrum in terms of why the elephants are doing this for the beetle. This is just what happens. So in terms of microbes, sometimes they're this kind of very loose food web of lots of organisms making things for lots of other, at kind of the next stage, making them for lots of other. But we have at least some rare examples where we have found kind of tight, specific interactions between pairs of microbes, even some where one organism is actually, one microbe is living inside or right on top of the other organism in an obligate fashion. There are some neat examples of a case, a pair of organisms where you have a photosynthetic microbe that sits, a whole bunch of them sit and ride a motile organism, and the motile one can't photosynthesize. The photosynthetic ones can't move, but collectively they can go back and forth in the proper light and oxygen gradients to be able to live together. And so there are some really interesting tight associations between microbes. The challenge for us microbiologists, though, is these are unfortunately exactly the kind of microbes that are the hardest to work with, because you can't just often get them to grow by themselves on a petri dish or in a flask. And so the very fact that they need their partners makes it really hard, because we want to be able to step in and do things with them. And as interesting as they are, fundamental questions are often some of the most difficult to address with these. And so in terms of thinking about then individual organisms and their community, that you can think of this reciprocally, in that you can ask, how does a community context affect the evolution of a given organism? The fact that their world is not just abiotic and static, it's changing. And similarly, as a given organism changes, as they evolve, they also affect their community properties. And these two things, of course, are linked. It's very much chicken and egg. There's not a start and a stop to it. And to address these sorts of questions, people certainly are taking this approach with natural systems, but they're often really, like I said, really difficult. Hard to grow, and if you can grow them, often hard to do genetics, hard to do the sorts of things that we would like to be able to test. And so the sort of approach that someone like myself, who's trained in molecular microbiology, and I like to be able to directly test experiments and have genetic control over what's going on, is to make a synthetic community. So I have kind of an arranged marriage of different partners, as you'll see. And so I set up a system where now they have to cooperate. And so rather than use kind of something that's interesting but extremely difficult to work with, we can set up something that's synthetic. And from the time we start these communities, that is now their real world. They will evolve. It's natural selection. But it's my kind of decision in terms of what that artificial world will be. And so let me start from the top and just mention kind of what the broad take-home messages are, and then I'll go through and lead to the sort of work that gets us there. And so one of the broad messages is that evolution is not something that takes billions of years. It happens. You can't help but have it happen because some variants are going to be more or less fit than others. And variants are always being made because replication is not perfect. And the sheer population size of microbes is phenomenal. Like if you think about the sort of stuff we do in the lab, we often will have, say, 1 billion microbes within a milliliter of fluid, right? Just in whatever that is, quarter teaspoon, something like that, right? And so even if mutations are incredibly rare, so about 1 in 500 bacterial babies will have a mutation. So you'd have to go back 500 great-great-grandmas before, on average, you'd expect to even have one thing have gone wrong. However, there's 1 billion of you. And so it means that there are 1 million mutations every generation. And so a few generations, you've saturated the entire genome. Every single mutation on average will have occurred. And so there's a tremendous possibility for different variants to occur, many of which won't matter, some of which will be bad, a decent portion of which will be bad, but some of which will happen to be better under the given circumstance that the organism now finds themselves. And the sort of evolution, particularly in the context of a community, it can happen really rapidly. And so as we'll see, the function of the community and the phenotype of the organisms can change in days, weeks, and months, not necessarily billions of years. The other aspect that I brought up is about spatial structure, that things become really different when you have neighbors, right? Our world is not just completely well mixed. And often one's relationships develop around who are around you and either you like or dislike. The same thing is true with microbes. The fact that you have spatial structure and it's not just completely well mixed matters tremendously, as we'll see. And then lastly, near the end, I'll throw in a little bit to suggest that we may be able to understand this from first principles. We're moving in this direction. It's a nascent sort of field, but there are types of models where you can take simply how individual organisms can be predicted to function and actually predict how the whole community would grow through space and time, and at least allow us to frame a hypothesis as to how, if I were to set up some given community and hit play, what would happen, something that we haven't been able to do in the past. So I'll have a few sections today. First, I'll talk a little bit about the model system, about experimental evolution, and about the two players here, E. coli and Salmonella. And I'll talk a little bit about the initial establishment of a consortia between these two microbes. And then I'll talk about the long-term evolution of these consortia, how we could make some predictions based upon what we saw from kind of the initial strains that could grow together that don't necessarily play out if you run things for a longer period of time. And then, like I said, I'll mention a little bit about how mathematical models can be used to address and tackle these sorts of questions. So the type of interaction we're going to have here is not just this one way, I happen to do something and release a compound and you happen to like to eat it, and so we have an interaction, but it's just kind of A feeding B. In this case, we're going to be talking about a situation where there's reciprocal exchange. In particular, where one partner can eat the food and can release products, but it needs something. It needs a nutrient from the other partner. And so the other partner, partner 2 here in red, needs a food source but has to provide back a nutrient, in this case an amino acid. And so the real question is, how does this play out if it's costly? If either one of these arrows, either the production of A or B, is costly, why would you do it? Right? Why don't you let some other genotype, some other strain of your own species do the work, and you just simply take the benefits? And so we can talk about that as being, quote, a cheater, right? Where you're reaping the benefits, but you're not putting in at least as much, or maybe not at all, relatively speaking. And so if we think about cooperation and what it takes to evolve, there are kind of two criteria. One is really simple, that whatever benefits you get from cooperation better outweigh the costs you put in, right? So if I'm going to feed you 100 molecules of this and I only get 1 molecule back and that's not nearly enough to pay for the 100 I just gave you, even in isolation, then that's a bad idea. But the problem is the world is not in isolation because other things can migrate in, or your own strain can mutate to other types. So you have to worry about if a defector shows up. So even if I'm a cooperator and I could make a living doing this cooperation, I have to worry about whether a cheater could show up and take away my ability to do what I'm doing. And so it turns out then that this difference between benefits and costs, you better have a better equation, the benefits minus costs for those who are actually doing something, has to be more than those who aren't. Otherwise, those who aren't will take over, right? And so how can you have this be true? Especially in a well-mixed world, because in a well-mixed world, the benefits are going to be the same, right? Everyone's going to get paid exactly the same, but the costs are borne by the individual. And so if the two B terms in this equation are exactly the same, even if something was free, even if it was absolutely free for me to give something to my partner, it's still not beneficial. It would simply be neutral. And if it was costly, it would only be deleterious. And you could never satisfy this inequality. So how can organisms, microbes, they're not these nice, fuzzy-looking things we saw on the first slide, make such a choice? So they could run around and look for partners that were specifically nice to them. And there are some examples of this. But there's a much easier way, and that's spatial structure. The idea that when microbes land somewhere, their neighbors are most likely like their brothers and sisters. And spatial structure sets up a situation where what you do benefits you, it affects you more than some strain that's further away. And so we can think about these two contrasting environments, right? Of our typical lab instantiation, where we have an Erlenmeyer flask spinning around in liquid. There's only one concentration of anything. There are no gradients. There's no locally more of this or more or less of this. Everything is well mixed, both the strains as well as whatever they're feeding each other. The other possibility, of course, is like a petri dish, or a rotten piece of food in your fridge, where there you've got localized little colonies of growth. And there, a colony can affect one that's close to it much more than one that's further away. And that makes a huge difference. Because if interactions are global, and so if we can kind of draw in the cooperator and defector, cooperator being the dark blue, the defector being in light blue, it doesn't really matter. Like they'll each be feeding the red species, and whatever the red species is giving back, they're both going to get the same. So it doesn't matter that the cooperator is giving more than the defector is. They're both going to get the same benefit. And so under these situations where there's no correlation between what you paid and what you get back, defection can win, and cheating will take over. But if it's structured, right, now if I'm right next to my partner species and I'm being really nice to them, I benefit, right? You can think of it as altruism, but it's in my own best interest if it's going to actually survive, because I'll be able to have a partner that grows well and gives me what I need. Whereas if you're not giving your partner very much of what they need, then you don't grow as well. So simply by having a structured world where the interactions are local, this leads to a correlation between the benefit and the costs, and now there's a chance for cooperation to win. Or I put a star next to it because it doesn't necessarily win. It might end up coexisting, where you can still have a slightly more complex world, kind of like ours is, where most people behave by the rules, but once you have a lot of people behaving by the rules, it becomes easier to be a cheater. So the specific system that we're going to talk about is one between E. coli and Salmonella. And now these are two organisms that don't really get a good rap in the press, I would say, right? I think if most people were to think of E. coli or Salmonella, you think of having to go to the doctor afterwards, etc. It turns out, E. coli is a very common part of most people's commensal flora in the gut. It's a minor component, 1.1%. It's not a major aspect of it, and most of them are perfectly fine. They don't do anything necessarily bad. Of course, there are variants that either pick up genes from other organisms and can become pathogenic, or can simply mutate the genes they already have and cause various infections. The other player, Salmonella. This happens to be typhimurium. This is why you wash your hands after cooking chicken, etc. Salmonella is not so common in mammals. It's more something that you have, in fish, in reptiles, in birds, etc. And so, for this sake, they don't necessarily evolve together. E. coli is more found within mammals. Like I said, Salmonella more in the guts of other organisms. But that's perfect. We want an arranged marriage. I don't want them to have already learned how to get together. I want to be able to see this happen in the lab. And so it's great that they're not necessarily already a co-evolved pair. And in particular, the E. coli is not just normal E. coli. It's actually one that can't make its own methionine, one of the 20 amino acids it needs to be able to make proteins. And so without that, they don't grow unless you give them methionine from the outside. And so what we're going to be asking for is for their partner to give them methionine. And so the food source, and I should mention, all of this was set up, this initial system was set up by a fantastic postdoc in my lab, Will Harcum. And this was work that he started at University of Texas, Austin, and we were doing some similar work, and he's since joined my lab, and we've gone forward with this. And what he set up was a situation where the food source can only be used by one of the organisms. So we're using lactose. This is the sugar that's in milk. Some of you might be intolerant to it, etc. This is one of the kind of defining characteristics, kind of typically, between E. coli and Salmonella. Salmonella don't tend to be able to use lactose. E. coli do. Again, this relates to being in a mammal gut, where lactose, of course, is relevant versus the gut of a chicken. And so when they grow on lactose, they do release some compounds. They release a little bit of acetate, for example, vinegar, etc. But they need methionine back. And so Salmonella, even though they can't grow on the lactose, they can eat some of these other carbon compounds made by E. coli, and they can give some back for the system to work. But it turned out that if you take just the normal Salmonella and put them together with E. coli, they just they don't happen to already release enough methionine for this to work very well. You get a tiny little piddly amount of growth, but not very much at all. And so what he did was to try to select for kind of this very first step. And so he mixed these two organisms together on a petri dish where there's only lactose to eat, so only one partner can eat, and yet only the Salmonella can make methionine for themselves. And so unless this exchange works out, they both have problems. And so mix them together, got just a little scum of growth, not very much. Wash them off, put some liquid down, scrape that around, shake it up, dilute it, spread it back out, just barely anything growing at all. He got frustrated, went backpacking, seek solace in nature. Came back, and there was some growth. And because of some indicators that we put some chemicals that tell us whether lactose is being utilized, we knew that this was real, that this was lactose utilization. And so you can imagine two scenarios. Well, three scenarios. You can imagine it being contamination, and the whole experiment is flawed. You can imagine the E. coli somehow got a workaround. They found some new way to make methionine, which seems somewhat improbable, and that wasn't the case. It turns out it was what we were hoping for, a Salmonella that emerged that could make methionine to feed its partner. And so it manifested as seeing this lactose utilization by the E. coli, but it was actually a Salmonella mutation that occurred randomly on this plate that allowed this to happen. And I could tell you more about the mutation and exactly what gene it's in, but that would be kind of a story for a whole nother night. The one thing I'll say is that you can repeat this experiment, and we've done it about 8 different times. You always get mutations in the same gene. And in fact, the same region of the same protein encoded by that gene. And another graduate student in the lab, Sarah Douglas, is tracing down trying to connect from the effect of the mutations upon this protein, exactly why that leads to this methionine production and exactly how that percolates through to the ecological phenotype of these two now being able to survive. But needless to say, we now have a strain that makes methionine, and it turns out it was costly. These methionine-producing strains are much slower. So if you just grow them on their own, they're about 40% slower because they're making so much methionine. And so we can ask now whether this is a good idea and under what environments. I already brought up this idea of liquid versus solid environments as being a way of testing whether or not spatial structure matters for the evolution of cooperation. And so what we can do is we can do kind of a three-way experiment. We can put in E. coli, the original Salmonella, which will take the food from E. coli but not make anything, and the new cooperative Salmonella. And we can ask both how the ratio of the Salmonella changes, as well as how do the numbers of E. coli and Salmonella in total change through time. And we can do that experiment both in a flask or on a plate. And so, first of all, if we look at the proportion of the Salmonella that are actually cooperators. So if we started in liquid, we tilted the odds entirely. We put 99.9% Salmonella that are cooperators, that make methionine for E. coli. So they're in there, the E. coli's in there, but there's a little bit of the non-producing Salmonella. And that little bit of Salmonella that takes the benefit but pays no cost, it's 40% better per generation because it's not slower. And so it rises up, 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 and eventually they take over the Salmonella population. And the cooperators die out. And on the x-axis is the number of transfers, how many times we would take the flask and restart a new one, restart a new one. All right, so the wild-type cheaters, they rapidly win because they get the same benefits and not paying the costs. What was the consequence? So if you look at the total number of cells, where kind of on the top is the Salmonella and the bottom is the E. coli, the Salmonella kind of held relatively steady for a while, but the E. coli start to flag once there aren't enough Salmonella to feed the methionine. And yet, but there's nothing to stop the impending crash, because under all scenarios, the cheater has an advantage over the producer, even as the populations are going extinct. And that's exactly what happened. Every single replicate population with very similar kinetics, there's actually little tiny error bars on there representing the variance across multiple experiments. They all go extinct at all exactly the same time. So this is selection-driven extinction. And sorry, Chuck, even though it's your birthday, but this is one thing that Darwin got wrong. The very few things he got wrong in the origin of species, but one of them he said that selection can't drive extinction. And in this sort of scenario, it actually can. When you lose this connection between a public good and you being able to benefit from it, you actually can have selection-driven extinction. In this case, not just of one species, but one species driving out the extinction of the whole community, both species. So how would this be different if you played it out on a Petri dish? Well, we had a feeling that this was going to work out much better, so we started the experiment in the other direction, right, where we started with just a small number of cooperators, mainly Salmonella that aren't doing anything, with their partner. And here, again, you're only going to grow, the Salmonella will only grow well if their partner is growing well. And so those who made methionine, their local ring of E. coli around them grew great, which means they grew great. And so each time we would wash the plate and transfer them again, their numbers would rise. But they don't actually go all the way to 100%. Because once there's enough of them to feed the E. coli pretty well, you can have kind of some hanger-oners, some cheaters of the Salmonella stick around. And it's not that the cooperative Salmonella want their competitors to stick around. They're not getting any benefit. But it's just a consequence, because they're feeding their neighbor well, their neighbor's feeding them back well, and it's just simply a consequence of their action. And because of this, the productivity of the populations in this case increase through time instead of going extinct. And so this is actually on the log scale in powers of 10. And so they actually, as the cooperators rise up in frequency, the numbers go up 100, 200-fold in terms of both the population of Salmonella and of E. coli. And so, this first example in liquid, some people may have heard of game theory, thinking about these things in different sort of ways. This, for those who have, this can be described as prisoner's dilemma. It's a situation where defection is just always the best thing to do. There's never a circumstance where it's actually better to cooperate. Where it's more complicated in a structured environment, it actually is a good idea to cooperate if there aren't many defectors around. But if there's a ton of cooperators around, being a defector is not so bad. So it's a little more complex of a world, perhaps a little more human in that way. And what was really exciting about this is, as much as this may seem like kind of an arbitrary little story, this is the first time ever in the lab that we got to witness cooperation emerge between two species de novo that was costly. And it required, again, in this case, the use of spatial structure. So this was the establishment of kind of these initial players. Now we've got a Salmonella that makes a ton of methionine and E. coli that needs it. And so we wanted to ask, how would this evolve in the longer term? And so we set up multiple populations. In this case now, instead of just doing one, we had a dozen populations on solid and a dozen populations in liquid. We ran them for 280 generations. This doesn't take so long. Something like about 3 months. Not so bad. And started with this methionine-producing Salmonella and this methionine-needing E. coli as ancestors. And we had some guesses based on what I just showed you, right? That there are some things that we might expect to occur. And so this is a weird, so this is experimental evolution where we're kind of playing this out for a longer period of time. And so anytime we do experimental evolution in the lab, we take kind of our particular starting strains and we let them rip. We let them go in whatever kind of environment. It can be flasks that I've got around here. In our experiment that I'm talking about now, they were on Petri dishes. You can evolve them forward. And under the conditions of these plates, what's really being selected for? What matters? What matters is how many cells can you make by the time it comes to transfer you, right? Will your colony have 1 million cells, or have 2 million cells, or have 10 million cells, et cetera? Because we're going to come with some liquid, scrape off the plate, dilute everything, and put a small number back down. And so if your type can make a bigger colony by the end of the time of growth, you'll begin to outnumber those that are making smaller colonies. And so that's fundamentally what's getting selected for in this environment. And the great thing about using microbes, and I should have perhaps mentioned this before, is unlike elephants or lions or things that might be a little bit more interesting to look at, you can keep them alive in the freezer. This is, you don't need some Austin Powers sort of deal going on here. And so we have a living fossil record. We have the original strains frozen away that we can revive, and then we have all of these populations through time, across the replicates. They're all saved, and they're all in our freezer. And so we did this a few years ago, but we can go back to whatever point in time we want and get things restarted. So not only do we know what happened, they're not just dead fossils, they're alive. And so we can actually compare the physiology of what's around today to what was there before. And so, all right, so what guesses would we have based upon what we saw from the first experiment? And so one is about E. coli. It's just simply making carbon as a byproduct. This is what it does when it eats lactose. It's not costly, and so since there's no kind of conflict over cost, perhaps it'll just evolve in that cost will never come up as an issue. Another possibility is that in a structured environment, what you do for your neighbor benefits you. And so you can imagine kind of this never-ending escalation. Right? It's like a never-ending chain of thank you cards. Thank you for that gift. Thank you for your thank you card. Thank you for the thank you card. So you can just build this up and up and up. And when will this ever stop? Right? Will productivity just go up, up, up? I mean, at some point, this has how nice you can be to your neighbor probably will stop. But perhaps it could go higher. And then the other thing is in liquid. All right, this should just be doomed, right? Where what you get is completely separated from what you do. There's no way. We'll start with an original pair that cooperates, but we showed before, if you put in just one defector, a small amount of defectors early on, these populations go extinct. And so we certainly expected that if we let these big populations go, given how many mutations would occur, that a cheater would arise and wipe out the population. And so these were kind of our guesses, kind of naive guesses that we had when we started, and we were correct about exactly none of them. Things change. So even the direct experiment you can play out with the original genotypes didn't necessarily play out because both of the partners changed enough that it completely changed kind of the game and what went on, which was really fascinating. I'm going to mainly talk about this first one, how E. coli initially really didn't care, just released food to its partner, but now that it has a partner, it actually cares about what it's feeding, and that's what we'll talk about. But the other aspects, no, there was not this never-ending thank you, thank you, thank you. If anything, the productivity went down, settled down just a little bit. And then also, our liquid populations that we thought we were just being kind of complete, doing this as a control, they all survived. And that, again, is a story for a whole nother night. We really thought that they would go extinct, and now we can run the exact same test. After they've evolved for a period of time, we can put in a cheater. All right, fine, you couldn't figure out how to cheat, we'll put one in, and watch you go extinct. And if you do that experiment with the original strains, you see this immediate crash out. You do it with the evolved populations, and darn it, they survive. We've yet to figure out exactly how, but the nice thing is it's there. We've got the strains, we can sequence their genomes, we can find out all the mutations that are involved, and piece this back together, which is lots of fun. So we'll get to this first one, E. coli. So one of the things we can do when we evolve these populations is we would check to see what's the ratio of E. coli to Salmonella. Are there now more of this partner than that partner, et cetera? And so we can plate them out on media that now, instead of lactose, which only one partner can eat, we actually have a rich medium. This is kind of like a soup broth, basically. And both strains can grow on their own, but we can distinguish them based on color. And so it might be a little hard to see, but there's both light blue and there's white colonies there in all cases. The light blue are E. coli, the white are Salmonella because of the use of a dye that distinguishes the two. But also you can probably see that each of these populations have these really, really dark blue. So like the other light blue is kind of the typical E. coli look, but these are ridiculously dark blue. So again, Will was concerned. This looks like contamination. All those populations, all this work, months of evolving them, this is probably down the drain. And so we checked, is this actually, did this strain actually come from our strain? Because we had particular markers, we would know what to look for, and so we could check, and it was. This had actually emerged. And so what actually causes this? And so the nice thing is then, like I said, we can use genome sequencing. Same way, like the Human Genome Project, except for ridiculously cheaper. Now we can sequence a genome of a bacterium for $200. I'm being a little conservative. It's probably a little bit less than that. And so you can get the entire sequence, compare it to their ancestor, and find what's changed. And there are relatively few changes, but one of them was in lactose utilization. I'll talk about exactly where. it was in a gene that encodes a protein called galactokinase, throws a phosphate group onto galactose. And it turns out, just to throw this in, again, there was a case of parallelism. Every population had dark blue colonies, and in all of them, they had a mutation in galactokinase. Different mutations. They weren't the same. So they emerged separately. But so even though they were random, the selective conditions were such that it was always advantageous to have this phenotype. For those of you who know about like the genetic code, where there are triplets of DNA that encode for each amino acid, all the mutations are also frame shifts, which mean a mutation that's not a multiple of 3. And so like some of these, for example, delete 2 nucleotides. And so that suddenly pulls your entire rest of the gene out of its original register, and the rest of the protein will be completely wrong. And interestingly, that's the type of mutation we even saw in all of these different cases. And in all of these cases, these E. coli strains don't grow on galactose. These seem to be mutations that are defective, that you're losing the function in this given gene. So what does this matter? And so we wanted to actually measure, well, what exactly is the E. coli feeding the Salmonella? And so we measured it in a couple of different ways. And one of the things, as we had expected, was the release of acetate. This is kind of a relatively common kind of overflow metabolite. Salmonella can grow on it. They don't really particularly like it. It's not a great thing to grow on, but they can. But there's a little bit of another compound called galactose, which I'll come back to. And so what we saw in those populations after a period of time, right, there were 2 different types of E. coli. Most of them were light blue and physically looked like their ancestor, even though they grow much better in these communities. But they have this light blue characteristics. They only release acetate. That's all they do. But these dark blue ones, they don't release acetate very much. They release galactose. And we can measure this via a particular kind of fancy machine. It looks just like a box, but there are fun things on the inside, and be able to see that this galactose is being made. And so where is that? In metabolism, I'll show you. It blocks, so lactose comes in the cell, and it gets cut into glucose, which is typical sugar, and galactose. So lactose is a disaccharide, 2 sugar molecules stuck together. It gets cut in half to a glucose and a galactose. The glucose goes right into central metabolism. The galactose goes through a few steps and then kind of joins up and has a similar fate. So normally you get 2 sugars for every worth per every lactose that you take in. In this case, these dark blue E. coli no longer can eat half their food. They chop it in half. They now take the front half, and they can't do anything because of the galactokinase mutation with the back half of the molecule, and they release it. They're giving away half of their food to their partner, which is awfully nice of them, perhaps even too nice of them. And so this was also costly, just like what we saw from the Salmonella. And so if we look, for example, we can measure growth of different E. coli, which I've labeled in either light blue or in dark blue, and on the x-axis, we've got how well do they grow alone. So if we give them lactose and give them methionine already in the media, so they don't have to feed their partner, you can ask how well they grow, and all the light blue ones grow much faster. 1 is kind of the original rate, and all the dark blue ones are relatively slow. That makes sense, so there's a cost. However, on the y-axis is how well does a community grow, where again 1 is kind of where the original community is, and the dark blue ones in general can allow their community to grow much better. So even though they are slower, they can feed their partner so much better that the net growth rate is actually faster. So even though I'm slowing myself down, I can help you so much that collectively we're better off than if I was a fast but somewhat more miserly version of E. coli. And so this is interesting. We have this diversification into these 2 different strategies, where some of the E. coli in this community have stuck with just basically making acetate and do that better. Some have completely changed, not just how much they make, but what they make for their partner. And so what can we think about this? There are a number of different things. And so one of which is, again, parallelism comes up, seeing this same outcome across different independent replicates. That happened for the Salmonella, with always hitting the exact same gene in methionine biosynthesis. It happened here, actually in 2 ways. One, that it was always galactokinase, so it was always the same gene, and in fact always frame shifts. But in the other way was kind of at the ecological level. In no case were the E. coli all dark blue. And in fact, the dark blue is a minority of every population. So it's a great idea if it's really rare, but by the time the dark blue rise up to a decent frequency, they are so helpful to their partner, then that the other E. coli can benefit enough that it ends up becoming, it comes to an equilibrium, to a balance, and thus you see coexistence between kind of the relatively less helpful E. coli and the relatively more helpful E. coli. Another major implication is that a simple little, boring, if you will, change within metabolism has tremendous implications by the time it percolates through to the community. One of my students was once asked during an exam, you're not selecting for cooperation, you're just selecting for methionine growth or for galactose production. And I'd say the answer to that is yes. That is exactly what's being selected for. But under this context, that is cooperation. When a legume that can grow in nitrogen-poor soil because it feeds carbon to the nitrogen-fixing bacteria that make little root balls in its roots, the bacteria are making nitrogen available, and the plant is making carbon available. We call that cooperation. It is also just selection for nitrogen and just selection for carbon production. But that's exactly kind of the basis of these sorts of metabolite-based exchanges. Another implication is kind of one of these chicken and egg questions. How do you ever get to a situation where two organisms are doing something costly for each other? Right? How do you get to that point? Because if just one of you is doing something costly, it makes no sense. And so the hypothesis had been this idea of kind of byproducts first, that at least if one of them just happens to do something for the other, there's an incentive for the second to do something for the first. So the first one doesn't necessarily have to change just by the second one doing something back, like what we saw from the Salmonella, making methionine back. You've closed the loop, and that's beneficial because you're already getting something back because that arrow to you is already pre-existing. But once the loop is closed, what's initially was just a byproduct, the E. coli just happened to make acetate and release it, now they actually need their partner. And some of them evolved to actually make a totally different compound that's much, much better for Salmonella growth than acetate is. And so we actually got to see kind of two steps in this process. The first link of costly cooperation and now kind of a reciprocation that's costly from the E. coli. And then the third is we usually think of being wasteful is not being a good thing, right? To take half your food and just spit out half of it seems like a pretty stupid idea. But maybe it's not if you have a partner, right? If you have a partner that you need something from, then maybe being wasteful is actually a good idea because you're getting something back. And frankly, when we look at natural microbes, they often do things that don't necessarily make sense if we treat them all by themselves. They often release compounds that don't make a ton of sense, and they often need so many things. They are so needy. And you wonder why. Why can't you just grow on your own? Why can't I just give you some food and some nitrogen, phosphorus, sulfur source, et cetera? Make everything for yourself. And a lot of microbes don't. You have to give them all these other things. And I think it's because what I showed on earlier slides, microbes don't live alone. If you always live in a community and you're always 0.001% of a community, somebody out there is going to be making these different things. Or maybe you even have a very specific relationship where somebody's making you a lot of something. And so kind of thinking about how natural organisms either release or need various sorts of compounds, I think we really need to think about it in the context of communities, particularly spatially structured communities that you would see in soil and things like dental plaque, stuff like that. So that's all kind of empirical. Can we ever try to understand this kind of from first principles? And so I have a fantastic collaborator, Daniel Segre, over at Boston University. And his lab has led the charge in terms of trying to model this. How could you actually just use math and computers to try to predict these sorts of outcomes? And it's every new computer program needs an acronym. This one is called COMETS, Computation of Microbial Ecosystems in Time and Space. It builds. There was some earlier work trying to model not just one organism's metabolism, but two. But they were unstructured and often would kind of impose that they already had to help each other rather than letting that emerge as a consequence. And so this COMETS, just in brief, it's kind of like a digital petri dish, that you have a model of each cell's metabolism. You can start them off in their world, and there's food stuck around, and you hit play. And off they go. They can use, we use their own metabolic pathways to predict, given my world, what should I be doing? And then we allow them to do that for a period of time, so they'll draw down their food, and so it'll diffuse in, and they spit things out, and those will diffuse out. And then every so often they reevaluate. All right, now my world is like this. Now what should I be doing? And their cell numbers go up, and you repeat. And so we can kind of iterate through this, assuming each organism is doing what they are doing for their own best interest, and ask whether things like cooperation would or would not emerge. And we use a type of models that ignore a whole bunch of detail and just take kind of the roadmap. It's kind of the Google Maps version of doing this sort of work, where you just ask what's possible, not the details of every given road, but just on the whole, think about metabolism. And so just extremely briefly, the basic idea is if we think about metabolism, which has all these various reactions, because the cells have to make a lot of things, right? You have to make protein, you have to make DNA, you have to make a membrane, then you got to make all the annoying kind of cofactors and this and that the enzymes that do those various things need. All these things, there's a lot of things that the cell needs to do. And you have to potentially use not just one food source, but many different food sources, et cetera. And so there's a whole bunch of possible detail there. And so the way that this works is basically you chuck out all that detail. You say, these are simply the reactions that can happen. And what's the goal of a cell? It's been said that the goal of an E. coli is to become 2 E. coli. And so all of this roadmap is to make more cells. And so you can propose then that the various things that need to be made, DNA, lipids, RNA, amino acids for proteins, et cetera, and the other various outputs, in the given ratio that you need those various things, go to make growth. There will be some byproducts, whether it be CO2 that you breathe off or other things like the acetate or galactose that we talked about. And instead of thinking about any of the details, this sort of model kind of sits back and says, well, what's all the possible ways the cell could be? What are all the possible ways I could kind of blast in food and it could flow through? And then you ask which would be the best. Let's assume that there's been selection to be the best. And so what would be the best way to allocate the flow of food down these different pathways? So you just turn it into kind of an optimal allocation. It's kind of like if I had a factory and I needed to know how many, I need to buy stuff. What's the ratio of wheels to chassis should I order? Well, if you're making cars, I'd suggest 4 to 1, right? That's what you're going to need. And so that's how you should be doing things. You're going to make semi-trucks? All right, fine. You're going to need a different ratio. And so based upon kind of your late downstream need, you can figure out kind of how should I be splitting things up to get to that point. So that's all. I won't get any more scary on the math. And so from this, we can make these sorts of various predictions. And what might we want to be able to do? So we can do things like ask, how much does it matter how close you are to your partner? Right? You would think in a mutualism it would be a great idea to be really close to your partner if they're doing good things for you. And so we can set up this experiment in the lab. We can set up E. coli and Salmonella at different distances. And so as before, the E. coli, because of a dye in the media, turns blue. The Salmonella stay white. And so we can either stick them right on top of each other or various distances that are further apart. And we can ask them, not surprisingly, the further they are apart, they're not growing as well, because much less of what they're actually doing gets to their partner, and conversely, much less of that actually gets back to you. And we can run the same thing in the model. And we can ask, how does it matter as space goes away? And the nice thing is the model can also predict things that are just becoming possible to measure. For example, it can predict kind of what would the concentrations of methionine on the top and galactose look like, as a function of space. Now that's actually a really hard question. Chemically, it's a hard question right now to measure how much chemicals are right here and 0.1 millimeter that way and 0.1 millimeter that way, et cetera. And there's actually just papers that are coming out having techniques to be able to do this sort of thing. So the nice thing is we can make predictions that we may not even be able to yet test, but we're moving down the road towards being able to do so. Now ultimately, so this experiment, let me flip back. So this experiment, these grids are like a half a centimeter. They're not so far apart. This was done with kind of a little device with lots of little pins sticking down, and you dip it into one thing and you plop it down. It's at a scale that kind of human manipulation is at. But microbes are a ton smaller, right? Like the first picture I showed you, they're right on top of each other. We're now not talking millimeters, we're talking micrometers, 1,000-fold smaller than a millimeter. And so how can we possibly do an experiment? Is it relevant to talk about spatial distance if we're going to talk about how well do I communicate with someone a mile away versus 10 miles away versus 100? All right, fine, 1 is closer than 100, but it may not be relevant if you need to be within striking distance. And so one way to do this is to do the same experiment but just really, really tiny. And so a good friend of mine, Nathaniel Cady, is a professor at University of Albany. He also actually happened to be a former roommate of mine in graduate school, and it's convenient to have friends in the right places. he can do exactly this sort of experiment. And so he and a graduate student in his lab, Rick Hines, have done a number of different experiments where they can print out cells at different distances. And so they can have different distances between them. You can print kind of different colors of dye, different species, and watch them grow through time. And there's various ways of doing this. One of them actually involves basically like a little tiny quill pen that they invented, where you can put the cells in. That's a particular color of your ink, and you can dip it down and make your different patterns. And so now we're just beginning to able to do this the sort of experiment that's moving towards relevance in an actual natural world. Now this is hard. This is new technology on the end of just printing it out. Trying to get the models to line up accurately with the experiments is something that's currently undergoing between the three of our various labs. But it's exciting because I think as difficult as this is, being able to actually measure what's going on here becomes harder and harder as your study system becomes smaller. But it's worthwhile because I think so much of the natural world happens at this kind of scale. We need to be able to move from kind of the more clunky, kind of scale that we're used to doing microbiology and move to their size of a playing field. And so, in terms of conclusions from this work, you can see that novel nutrient linkages can emerge incredibly quickly within communities. And so even though we see this great complexity of things going on, it didn't necessarily take eons to emerge. These sorts of things can happen quite quickly. Spatial structure is absolutely important. Because it has this, this fundamental property of making things be local rather than being global, and allows the benefits of things to correlate with the costs, because you're paying the costs yourself as a given cell. And then lastly, these models that just use kind of what can happen within a cell offer kind of a starting place where we can just simply take a genome sequence of an organism, try to make a model out of that, and try to be able to make kind of initial predictions that we can test. If they're wrong, they're wrong, but at least we can finally make an actual reasonable prediction of what might be going on if you had a microbial community and you let it rip in a particular condition over space and time. And so this has been a collaborative work. I have many excellent collaborators, notably Daniel Segre at Boston University and Nate Cady at University of Albany. Money, which is an important part of this picture, has come from the Department of Energy, because they're, of course, interested in how these sorts of processes affect a number of different things, from the production of biofuels in terms of kind of an above-ground synthetic process, to thinking about how microbial communities that do things like detoxify toxic metals underneath the ground, which is what our grant is also about, this is a process that's not just accomplished by single organisms, but in the context of communities. And then I have a fantastic lab. This is standing in front of a rhino statue that was dressed up as an E. coli. There's a flagellum tail on it. There's a virus that you can not see very much. And Will Harcombe's got his name in yellow. He again is the postdoc that did a large amount of the work that I talked about. I'd be happy to take any questions.
So how do you sequence genomes in the lab?
Yeah, that's a great question. So it's changed tremendously. Initially, the way sequencing was done is you'd kind of start at each end of a molecule that you would want to sequence. You'd sequence in a bit because you didn't know what was there. And then when you got to the end, you'd design kind of a new piece and sequence forward, et cetera. And, and so you would, like with the idea that you would read DNA like you would read a book, right? You start at one end and just kind of go through. That's not how we do it anymore. The way that it's done now is basically, instead of, if you want to read a book, go buy 100 of them, put them in a blender, take them into little tiny shreds, and sequence just a ton of them, because it's so much easier. So you sequence a ton of shreds, and each shred might be a sentence long, or a fragment of this sentence, et cetera. And then you try to line all the sentences up and figure out where the book is. And obviously, the is going to show up in many different places, but hopefully, 470 years ago, et cetera, will only show up once, and you can kind of line things up. The technicalities of how you do the genome sequencing has also changed tremendously. A lot of it, in principle, it's still based upon the idea that you actually use the enzyme DNA polymerase itself that can take one strand of DNA and then stick on the appropriate matching base pair. So across from every G is a C, across from every A is a T. And so you need to somehow be able to see that happen. And the way that people used to do it is you would start at one end and let it go forward, and you'd give them A, G, C, and T. And in a given reaction, you'd put a little bit of T that wasn't quite right it was a tea that once it went in, you couldn't keep going, and it was also radioactive. That's how they did, at least initially. And so it would run forward, and every so often it would stop at that tea and get stuck, and it would be radioactive and only that long. And sometimes it would go further and get stuck at that point. It would be, and only the kind of the messed up tea would be radioactive. And so you'd get kind of, you could take that reaction then, run it out on a gel where you use electricity to pull the DNA through, and little molecules can run through this gel faster than big ones. And so you could separate out these different stretches and say, all right, I've got T's kind of like here, 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 there, there. You do the same thing for G and C. Pain in the butt. You can do this. It's hard. You'd use these huge glass plates. It's technically really challenging. The DNA, the quality is pretty horrible. Now the stuff is done in like little microfluidic devices where you have just a ton of little DNA molecules that you can kind of do this thing just kind of massively in parallel in a few different sorts of ways. And so instead of getting a very small number of things, like there's a paper that when I teach that says, it has this bold statement, one day a single lab will be able to sequence 100 genes in a year. Well, now we can sequence, like 100 genomes for like nothing, and so like it's become tremendously easier. Yeah.
What would happen if a microbe was infected by a virus?
Yeah, so microbes also have to worry about catching the flu. So there are, just like we have viruses, bacteria certainly do also. They're called phage, but bacterial viruses do the same sort of thing. They attach onto the cell, they inject their DNA in, and then there's actually kind of two types of viruses. Some of them will immediately kind of take over that cell, and instead of having the cell make its own proteins, they suddenly make only viral proteins. And they'll make lots of kind of viral particles, and then the virus, kind of at the end of all of this, will make a protein that will pop the cell, and then they all go out, and suddenly now there's lots of little viruses, and out they go to do the same thing. Others are a little more sneaky. They'll occasionally slip into the host genome, and they basically, like, as long as when they're in the genome, to some extent, the bacterium would be asymptomatic. They can still grow and increase in number, and then later, they're still there. They can be released again. And so that's just like how chicken pox, you can get chicken pox, and then later you can end up getting shingles, or like AIDS can go in, be, in that case through reverse transcription, insert into the genome and much, much, much later come out of a given cell.